In Figure 1, we demonstrate that the NAMPT-mediated NAD biosynthesis pathway is essential for rod photoreceptors, which are the light-sensitive cells in the back of the eye that are responsible for vision in dim light. Figure 1a confirms that there is a significant reduction in NAMPT expression in the retinas of mice that have NAMPT deleted selectively from rod photoreceptors with the classic Krelok system. We next measured the retinal NAD levels of these mice. As you can see in Figure 1b, we found that retinas from rod-specific NAMPT conditional knockouts had a significant reduction in retinal NAD levels. We also measured the NAD levels of retinal isolates that had been enriched for rods. As shown in Figure 1c, enriching for rods increased the magnitude of the NAD deficiency in rod-specific NAMPT conditional knockouts. These findings suggest that the NAD deficiency we observed is predominantly specific to the rods. Next, we wanted to test whether this reduction in retinal NAD was accompanied by retinal degeneration. Figure 1d shows that by 6 weeks, rod-specific NAMPT conditional knockouts had clear evidence of degeneration on fundus biomicroscopy. This appearance of the back of the eye is similar to that which clinicians observe in patients with severe retinal degenerative diseases, such as retinitis pigmentosa. Consistent with this fundus appearance, Figure 1e shows clear evidence of retinal degeneration in rod-specific NAMPT conditional knockouts by histology. In particular, the arrows shown highlight the outer nuclear layer of the retina, which is made up of nuclei of the photoreceptors. It is obvious that this layer is noticeably atrophic in the rod-specific NAMPT conditional knockouts, indicating massive rod photoreceptor death. Next, we wanted to test whether these anatomic abnormalities were associated with the retinal dysfunction by using a technique called electroretinography, or ERG. In short, this technique allows us to measure the response of the retina to different flashes of light in anesthetized mice. As you can see in Figures 1f, 1g, and 1h, the rod-specific NAMPT conditional knockouts had almost no retinal response to any of the different light flashes, indicating severe retinal dysfunction. Finally, we tested visual acuity by measuring the optokinetic reflex. As you can see in Figure 1i, we found that rod-specific NAMPT conditional knockouts also had reduced visual acuity. Taken together, these findings all point to the importance of NAMPT-mediated NAD biosynthesis for both the survival and the function of rod photoreceptors.